G'day. This is Sandra again. Welcome to lesson 5.4. We'll talk about grazing on fungi. So we are going to talk about the little animals, the little creatures that love to eat fungi and how that affects fungal diversity, how the intensity of their grazing affects fungi and fungal diversity, and their food selection choices, what types of fungi some of these creatures like to eat, and how that affects the fungal populations and diversity, and how by doing so, by feeding on fungi, these animals actually distribute fungi throughout the ecosystem, distribute their spores or the mycelium, and also how some of them actually like to farm fungi and consume fungi by feeding the fungi certain foods. In all this, we'll also look at how that affects nutrient cycling in the ecosystem and the dynamics of the ecosystem as a result of this grazing behavior on fungi. Okay, so let's jump into lesson 5.4. Animals that graze on fungi, anyone that likes to eat fungi, is termed mycophagous. So that means that they just like to consume fungi. And for a lot of these animals, such as certain arthropods, animals that have jointed appendages, so insects, but also animals that have more than 6 legs, and some of the worms and mollusks, so some snails and nematodes, literally only eat fungi, prefer fungi, and even some mammals prefer to just eat fungi over anything else. So they are the mycophagous animals. The diversity of these invertebrates or these arthropods and the community composition of these animals is a key factor in driving fungal functioning and how fungi service the ecosystem and cycle nutrients as a result of being fed upon by these organisms that like to consume fungi. Some of these organisms ingest hyphae, and some will eat the mushroom and therefore disperse the spores of those particular species of fungi that they prefer to eat. And this really influences fungal mediated nutrient distribution throughout the ecosystem. For example, they can damage a fungal hypha, and when this happens, there's nutrient leakage that occurs in that spot where the organism has fed on the fungal species, on the hyphae that it wanted to consume. Also, by consuming fungi, they will excrete, obviously, the nutrients through their feces that they consumed via the fungi. And remember, fungi are extremely nutrient rich. So every time an organism consumes fungi, it will actually excrete way more nutrient than the organism actually requires, because that organism will always have less nutrient density compared to the fungal food it consumes. And this, of course, then leads to nutrient mineralization. And remember, what this means is that the organic nutrient that was present in the fungi is now returned to mineral form. And what this means is that the plants can feed directly on those mineralized nutrients. So it becomes plant food immediately as the organism consumes the fungi and transforms or mineralizes that nutrient. This also affects carbon dioxide fluxes within the ecosystem. So how carbon is either released into the atmosphere depending on the feeding behavior and which organism does that, versus how much of it is incorporated into the soil organic carbon component. So this grazing mechanism is extremely important to nutrient and carbon fluxes and nutrient cycling within the ecosystem. There's also preferential grazing and its intensity. So how certain organisms prefer to eat certain types of fungi and how intense the grazing behavior is on those particular species that then causes selective pressure, and therefore fungal succession and community composition is influenced within the ecosystem based on these preferential grazing methods and the intensity of that grazing. So in the diagram on the right describes the effects of invertebrates grazing on fungi and how that affects fungal nutrient distribution, mycelial physiology and nutrient cycling, and fungal community composition and diversity. So there are ecological aspects of fungal grazers and how they interact with fungi, and functional aspects. The ecological aspects relate to population dynamics of both the grazer and the fungi, and community composition of those two groups of organisms as well. And then there are functional aspects, and that relates to nutrient distribution, depending on whether spores are consumed or mycelium is consumed, and nutrient cycling, so respiration rates, how the fungi respire carbon dioxide as a result of being consumed, and also depending on how enzymal activities within the two different groups of organisms affects the nutrient cycling in the ecosystem. So these are the functional and ecological aspects of fungus grazer interactions. And majority of this lesson is actually based on a review called Functional and Ecological Consequences of Saprotrophic Fungus Grazer Interactions that you may wish to have a read of. So let's look at how the various organisms feed on fungi and how that feeding behavior influences the fungal populations and fungal species. So grazing intensity is very much dependent on invertebrate abundance, how many different species there are within the ecosystem grazing on fungi, and also how many of each of these species exist in that ecosystem. And that is also dependent on the species of invertebrates and the functional groups of the various fungi that support those creatures consuming those fungi. So basically, it really depends on the types of fungi present in the ecosystem and the abundance of those species and how that attracts the different kinds of organisms that then feed upon those fungal species. So we can look at it from a perspective of the functional groups, the different organisms, say snails, being predominant in particular environment because they have a specific way of feeding on fungi, versus species diversity. So how many different species of snails are there and how that affects mycelial functioning. And there are various functional groups, obviously, as we described earlier. Snails, you could have earthworms, you can have different types of arthropods. So it all just depends. But the rule is that high intensity grazing decreases fungal growth and activity, and greater invertebrate abundance decreases fungal biomass. So that makes sense, right? Because the more organisms you have feeding on fungi, of course, that's going to reduce the fungal population's fungal biomass. And also nutrient translocation is affected, and mineralization of those nutrients and carbon dioxide release into the atmosphere because of reduced fungal biomass in the first place by an overpopulation of invertebrates feeding on fungi. Low intensity grazing, on the other hand, increases fungal nutrient mineralization and plant growth because low intensity grazing actually stimulates fungal growth. Decreased fauna abundance means increased mycelial respiration because fungi are now growing, their growth is stimulated, so there's more respiration occurring, and soil nutrient availability as a result. Because of this lower intensity feeding, fungi are continually able to grow, and therefore that increases nutrient availability in the ecosystem. Larger animals such as earthworms, isopods, millipedes are the main regulators of mycelial activity even at low abundance. Because these animals are larger, they're actually able to sever entire connections between fungal colonies, so fungal cords. They can consume large amounts of mycelium, large amounts of hyphae, chunks being taken out, and therefore they really have an impact on how these fungi recover or don't recover in terms of whether they will grow well, depending on the species that's being grazed as well, and how that species responds to these organisms taking large chunks of the fungal colonies out of the system. So it is a bottom-up effect where fungal resources in the first place drive invertebrate populations and grazing intensities. So it really depends on the species of fungi as well as the organisms that feed on them and the resilience of the system and how much it can come back after being grazed. But really it's the fungi obviously being present that attract the types of organisms that eat them and influencing grazing intensity as a result. On the right, you can see an image where different grazing intensities are shown and how the two different species, Hypholoma fasculara and Phanerochaete velutina, respond to grazing. We can see from this image that when the fungi are ungrazed in image A and B, that's the extent of their colonies that they form growing on a piece of wood. Then when they are grazed by springtails or calembola, their growth is actually increased. The fungal growth increases as a result of low intensity grazing in both the species. However, when they are intensely grazed, where the intensity is increased, the fungal growth is actually reduced. They cannot recover in time when they are intensively grazed by the springtails. So this is a really important thing to think about in terms of balance within the ecosystem. So obviously we need species to graze. We need organisms to feed on fungi in order to increase fungal growth and therefore nutrient cycling and all the other ecosystem services that fungi provide. But when there's an imbalance in terms of reduction of fungal diversity in the ecosystem, say from climate change impacts, organisms will just feed on what they can and actually prevent nutrient cycling by preventing fungi proliferating. And of course, then the organism group will eventually die out because there will be no more food left. So it just shows how important it is to keep a balance between all the species within the ecosystem, starting from plants and fungi, to then provide this abundance in ecosystems in terms of food so that every organism can feed and feed in the right amount in order to keep proliferating the species of fungi and therefore support plant growth and diversity in return as well. So it's a very important ecological understanding that we have to develop in order to make sure that our ecosystems function properly. And obviously, in monoculture systems, this would completely fail. And this is why we have such food production problems. And the same goes for our ecosystems in the environment. We need that biodiversity to be maintained. That means that all the things function together, both the climatic conditions, moisture being maintained so that we can maintain diversity within the ecosystem. When all of those things fail, starting with climate change that affects moisture content in the soil and temperature, this already starts to have an impact on reduction of all species. Fungi don't exist in dry climates, and that's not entirely true because there are some species that do and grow in the desert, but majority of these fungi require moisture. So the moment we reduce that, we reduce the nutrient cycling for the plants, and that's because we're also reducing the organism numbers and diversity that can feed on these fungi to support plant growth, to mineralize nutrients from fungal sources. It's also very important to mention that these organisms, these fungal feeding organisms, form an essential food source for larger organisms that feed upon them. So when we don't have the fungal biodiversity that these organisms rely on, the fungal grazers rely on, and those organisms cannot exist, obviously, then the next trophic level, the next level of feeding in the food web can no longer be supported either. So it is just a flow-on effect from how we impact the environment and reduce this fungal biomass. All the organisms that rely on that fungal biomass can no longer exist in the balance that they are required in order to feed the next trophic level or the next level in the food web and so on. So you can see how important this actually is, how incredibly vital it is that we have everything present in abundance, in a state of diversity, immense biodiversity, in order to maintain above ground biomass of organisms. And that includes us, of course, because we rely so much on ecosystems being healthy for so many different things. For our entire livelihood, our life is dependent on ecosystems functioning so we can just even breathe. The other aspect of the dynamics between fungi and their grazers is the dispersal mechanism that fungal grazers provide, and also the capacity of certain species to farm fungi, or the reliance of some fungi on the species that farm fungi as their food source. So vectors are organisms that assist in dispersal of fungal spores or seeds in plants across the ecosystems. And this might be a passive dispersal where there's external adherence of fungal spores to the body of the organism. By chance, it's feeding on the fungi and then its mouth gets covered in spores and then it goes and inoculates a piece of wood as it goes and feeds somewhere else or goes and cleans itself. Another possibility is through gut passage. So when the organism actually consumes the fungi for the nutrients, say a mushroom, it will then disperse the spores by the spores going through the gut of the organism, and that's being excreted through the feces. Things like orbited mites, earthworms, collembola, which are springtails, and chytrids, which are small worms, have an effect that is both vertical and horizontal on fungal propagule redistribution. That's because they move both horizontally across ecosystems and vertically in the ground. Like, for example, earthworms, they can deposit their feces on the soil or in the soil as they burrow. So this is just dependent on the species, but that's the extent that some organisms have on fungal dispersal. Active dispersal is when specialized sacs on or in the invertebrate itself, on the body of the organism, are designed for gathering fungal propagules, fungal spores, where it's an adaptation of the organism to live with fungi in a mutualistic symbiosis. And then this organism has these specialized features on the body in order to actually actively, on purpose, disperse fungal spores and create more fungal biomass. And examples of that are termites. For example, the higher termites have a relationship with fungi called termitomyces, where the fungi cannot actually exist in the ecosystem without the termites farming the fungi. So these fungi only exist because the termites actually farm them. They create special conditions for the fungi to actually exist in. There's special humidity, they feed them special foods, and then the fungi transform that food for the termites to then consume the fungi as a nutrient source. Another example are attine ants that farm different species of fungi as well. Even certain wood wasp families have a relationship with specific species of fungi and farm those fungi for the purpose of gaining nutrients from the fungi. So there are very many different relationships between invertebrates and fungi that serve in the dispersal of fungal spores and distribution of fungi across ecosystems. And these relationships can be very specific as to like even going to the level of farming these specific fungal species, but all of that serves the purpose of fungal distribution and nutrient distribution within the ecosystem. So it actually drives ecosystem progress and it moves ecosystems through succession as a result. And of course, there is selectivity within organisms' preferences as to which fungi they prefer to consume. So, for example, different species of earthworms can actually reduce or prevent germination of several basidiomycete and ascomycete spores. And this depends on the species of fungi that they consume. Whereas some fungi of certain ascomycetes, their spore germination relies or is increased by certain worm species, and this gives them a selective advantage because as the fungal spores move through the worm's gut, they actually get stimulated into germination, and then when they are deposited via the feces of the worm, they actually are able to then grow as a result of being consumed in the first place. There are also certain termites and fly larvae that increase spore germinations of different types of species of fungi, and the germination of Ganoderma spores of the genus Ganoderma can only occur via passage through the invertebrate gut. So without being stimulated by being consumed, some of these species of Ganoderma cannot actually even grow. So without these organisms, without the invertebrates present that feed on these Ganoderma species, the life cycle ends. There's absolute requirement that these organisms exist in the ecosystem for the Ganoderma to even exist. So this is very interesting, and there are all these specialized relationships, and they're very complex, and it all has an effect. And once again, this biodiversity is so essential to preserve and so essential to create from the soil up in order to sustain these ecosystem functions that are so interdependent on one another in terms of how the species interact and what actually happens and what eventuates and what sort of an ecosystem is developed based on all these interactions that are happening and that are essential, mutualistic, and very, very specific. So biodiversity is the key as always. We also have to look at the relationship between grazers and mycorrhizal fungi. This relationship is parallel with that of saprotrophic systems, in that spore ingestion can either have a positive or negative effect on the distribution of the fungal species. So this depends on the grazer-fungal combination that occurs. Severing of hyphae actually reduces growth and nutrient translocation in mycorrhizal fungi. So it's better that these fungi are actually not fed upon, given that plants very much rely on those nutrients being delivered directly to the plant via these mycorrhizal networks and the mycorrhizal hyphae. And there can be either a positive or negative effect on fungal activity, leading to nutrient mineralization and primary productivity. High intensity grazing reduces mycorrhizal activity versus low intensity grazing that increases growth and increases plant productivity. So these are the parallels between grazing on saprotrophic fungi and mycorrhizal fungi. However, the good news is that invertebrates always prefer saprotrophic versus mycorrhizal fungi as food, and it allows the invertebrates to have a greater reproductive success as a result of feeding on saprotrophic fungi. So they will always prefer to eat those. And in complex multi-species environments, the fauna actually increases mycorrhizal growth, decreasing competing or inhibitory or even pathogenic fungi. So in very healthy ecosystems, through the dynamics of grazers on different species of fungi, such as plant pathogens, for example, or fungal species that are not contributing to plant growth, in these systems, there is a support of mycorrhizal growth by fungal grazers. So it's a wonderful example of, once again, the biodiverse ecosystem that is complex and how that always leads to positive interactions for all species to perform their roles and serve the ecosystem. So this is actually the end of lesson 5.4. And in the next lesson, lesson 5.5, we'll look at fungi and fire, the effects of fire on fungi, and we'll discuss some of the research that has been done on that relationship and how fire and fire intensity actually affects fungal populations and ecosystems as a result of the effect of fire on fungi. Okay, so you'll be hearing from me in lesson 5.5. See you then.